interestingly, the technology that you're talking about that Viome uses, where did it come from?
So this was developed at Los Alamos National Laboratory. It was funded by the US government in order to develop one capability that can identify every single living organism in any sample. And the only way to do that is really to analyze RNA. And so the government didn't want to study separate organisms and try to guess as to what might be in a particular sample. You simply want to have one test, and in that one test, you can identify every single living organism on this planet.
However, it's not about just identifying the organism. The purpose really was to find out what they're actually producing, because this is really what they needed to know to find out if something bad happens, what is making people sick.
Exactly. So you've heard of these strains of bacteria that can cause disease, but they also live on us or inside of us. So for example, Staph aureus is a known pathogen. However, Staph aureus lives on us as a commensal, as a friendly organism, but sometimes it turns into a pathogenic organism. So if you look at this organism from the DNA perspective, it's identical. It's the exact same organism. You can sequence it 1,000,000 times and you'll get the exact same data back. However, from the RNA perspective, when it's a friendly and commensal organism, it's expressing certain genes that enable it to live in our throats, for example, as a friend. But when it starts expressing these virulent genes, that's when it becomes pathogenic. And the only difference between a pathogenic Staph aureus and a commensal Staph aureus is in gene expression.